Continuation. Next, oncogenes that encode protein that affect the epigenetic state of chromatin. Earlier, you learned two types of oncogenes, which are nothing else but oncogenes that encode growth factor or their receptors. Then oncogenes that encode cytoplasmic protein kinases. Later, we learned one more type, that is oncogenes that encode transcription factors. Oncogenes that encode growth factors or their receptors, oncogenes that encode cytoplasmic protein kinase, then oncogenes that encode transcription factors. Later, we are going to learn oncogenes that encode proteins that affect the epigenetic state of chromatin. Two of the most important factors in determining the epigenetic state of chromatin are whether particular sites in the DNA of gene promoters whether particular sites in the DNA of gene promoters are methylated or not, and the particular modifications present in the tails of certain core histones within the nucleosomes of these same gene promoters. DNA methylation tends to silence gene, whereas histone modifications may either activate or repress gene transcription. Recent studies have indicated that a number of oncogenes encode proteins that affect DNA methylation or histone modification. These include DNA methyltransferases, histone acetylases and deacetylases, histone methyltransferases and demethylases, proteins present within chromatin remodeling complexes. Mutations in any of these classes of genes can promote tumorigenesis by increasing or decreasing transcription of genes involved in the various signaling and regulatory pathways that affect cell proliferation, survival, migration, etc. To cite just one example, acute myeloid leukemia is classified and characterized by recurrent mutations in DNMT3A gene, whose product is involved in maintaining DNA methylation patterns during DNA replication. A reduction in the level of DNA methylation could lead to increased movement of transposable elements, which could cause genetic instability as well as increased transcription of certain proto-oncogenes. Conversely, an increase in the level of DNA methylation of the promoter region of tumor suppressor gene is known to silence the expression of genes that exert a key inhibitory influence on tumorigenesis. Then oncogenes that encode metabolic enzymes. It was mentioned that tumor cells depend much more on glycolysis than do normal cells. This is only one of a number of major differences in metabolism between normal and tumor cells. Another difference, which was one of the surprising discoveries to emerge from genome sequencing studies, was the repeated sequence of mutations in the TCA cycle enzyme. Mutations in the isocitrate dehydrogenase, IDH1 and IDH2 in the tumor cells of patients with glioblastoma or brain cancer, and acute myeloid leukemia. These mutations cause these enzymes to lose its normal activity of converting isocitrate to alpha ketoglutarate and instead convert the substance to an abnormal metabolite called 2-hydroxyglutarate, which accumulates to high levels of the tumor. The elevated levels of 2-HG have an impact on a number of processes involved, including histone demethylation and DNA methylation. It is proposed that the disruption of these epigenetic processes would likely result in the aberrant regulation of gene expression within tumor cells. Then oncogenes that encode proteins that affect apoptosis. Apoptosis is one of the body's key mechanisms to rid itself of tumor cells in an early stage in their progression towards malignancy. Consequently, an alteration that diminishes cells' ability to self-destruct would be expected to increase the likelihood of the cell giving rise to a tumor. This is evident in the previous discussion, which was done with the help of the previous video of P13K to PKB pathway in cell survival and tumorigenesis. With this in mind, it is not surprising that both PI3K and PKB are encoded by documented oncogenes. The oncogene most closely linked to apoptosis is BCL2, which encodes a membrane-bound protein that inhibits apoptosis. The role of BCL2 in apoptosis is more clearly revealed in the phenotypes of knockout mice that are lacking a BCL2 gene. Once formed, the lymphoid tissues of these mice undergo dramatic regression as the result of widespread apoptosis. Like MYC, the product of BCL2 gene become oncogenic when it is expressed as a higher than normal levels, as can occur when the gene is translocated to an abnormal site on the chromosome. Certain human lymphoid cancers called follicular B cell lymphomas are correlated with the translocation of the BCL2 gene next to gene that codes for the heavy chain of antibody molecule. It is suggested that overexpression of BCL2 gene leads to the suppression of apoptosis in lymphoid tissues, allowing abnormal cells to proliferate to form lymphoid tumors. The BCL2 gene may also play a role in reducing the effectiveness of chemotherapy by keeping tumor cells alive and proliferating despite damage by the drug treatment. Over the past few pages, we have discussed a number of the most important tumor suppressors and oncogenes involved in tumorigenesis. Tumor suppression and tumor suppressor pathways are shown, and next topic is the mutator phenotype. Mutant genes involved in DNA repair. If one considers cancer as a disease that results from alterations in DNA of somatic cells, then it follows that any activity that increases the frequency of genetic mutations is likely to increase the risk of developing cancer. Nucleotides that become chemically altered or nucleotides that are incorporated incorrectly during replication are selectively removed from the DNA strand by DNA repair. DNA repair processes require the cooperative efforts of substantial number of proteins, including proteins that recognize the lesion, remove a portion of the strand containing the lesion, and replace the missing segment with complementary nucleotides. If any of these proteins are defective, the affected cell can be expanded to display an abnormally high mutation rate, which is described as a mutator phenotype. Cells with a mutator phenotype are likely to incur secondary mutations in both the tumor suppressor genes and oncogenes, which increase the risk of becoming malignant. MicroRNAs, a new player in the genetics of cancer. MicroRNAs are tiny regulatory RNAs that negatively regulate the expression of target mRNAs. Given that cancers arise as a result of abnormal gene expression, it would not be surprising to discover that miRNA somehow were involved in tumorigenesis. In 2002, it was reported that the locus that encodes two microRNAs, MIR15A and MIR16, was later detected on underexpressed in most cases of chronic myeloid leukemia. It was subsequently shown that these two miRNAs act to inhibit expression of the mRNA that encode the anti-apoptotic protein BCL2, known as proto-oncogene. In the absence of the miRNA, the oncogenic BCL2 protein is overexpressed, which promote development of leukemia. Because these miRNA act to inhibit tumorigenesis, they can be thought of as tumor suppressors. And the leukemia cells lacking MIR15A and MIR16 were genetically engineered to re-express these RNAs. They underwent apoptosis as could be expected if a missing tumor suppressor activity was restored. The locus that encodes MIR15A and MIR60 was deleted in other types of cancer, suggesting it was widespread importance in tumor suppression. The expression of two of the most important human oncogenes, RAS and MYC, have also been shown to be inhibited by the mRNA, namely LECT7, which was the first mRNA to be discovered. Some miRNAs act more like oncogenes than tumor suppressors. One specific cluster of miRNA gene, for example, is overexpressed during the formation of certain human lymphomas. Overexpression of these miRNAs can occur because the gene cluster encoding them is present in increased number, amplified in the tumor cells, or it can occur because the gene cluster is excessively transcribed by the result of overly active transcription factors, including MYC. And mice are genetically engineered to overexpress this particular miRNAs. The animals develop lymphomas, as would be predicted if the gene encoding them were acting as oncogenes. Overexpression can be oncogenic when the miRNA target the mRNA of key tumor suppressor genes such as TP53. This appears to be the case in many neuroblastomas where the TP53 gene is not mutated, but it is not it's not expressed at normal levels. The abnormal expression of miRNA has also been implicated as a cause of tumor cell invasion, illness, and metastasis, which elevates the interest in the RNA to an even higher level. It is not yet clear how important mRNAs are in the overall occurrence of human cancer, but a number of microarray studies have survey large number of these tiny granular RNA suggest that most human cancers have a characteristic mRNA expression profile, just as they have a characteristic mRNA expression profile. Studies suggest that mRNA expression profiles may serve as sensitive and accurate biomarkers to identify the exact type of tumor a person is suffering from and the best avenue of treatment. The studies also suggest that mRNA may serve as a potential target for anti-cancer therapeutics. It may be possible, for example, to treat patients with specific RNA that act as mRNA sponges. Therapeutic RNA of this class would possess sequences that were complementary to oncogenic mRNA and would act to bind and sequence such mRNA, thereby blocking their carcinogenic activity. In the cancer genome, all cancers arise as the result of genetic alteration, as is evident from the previous discussion. The genes involved in tumorigenesis constitute a specific subset of genomes whose products are involved in such activities as the progression of a cell through the cell cycle, addition of a cell to its neighbors, apoptosis, and repair of DNA damage. Taken together, more than 350 different genes have been identified as cancer genes, whereas genes that are thought to have some causal role in development of at least one type of malignancy. Over the past few years, a concerted effort has been made to determine which of these genes are altered, either by point mutation, translocation, detection, and duplication in various types of tumors. This effect has been aided in recent advances in DNA sequencing that allow researchers to determine the nucleotide sequences of specific regions of genome much more rapidly and inexpensively than ever before. It was hoped that most types of cancers would be characterized by alterations in a relatively small number of genes. It has been known for a quite long while. For example, that a large percentage of melanomas exhibit mutant BRAF genes and a large percentage of colorectal cancers exhibit mutant APC genes. Similarly, the various types of leukemias are characterized by specific translocations as exemplified by the BCR-ABL translocation and CML. In all of these cases, these mutations occur at an early stage in a tumor development and is thought to be critically important in turning the cell toward an eventual malignant state. However, results from the initial studies of cancer genomes suggest that the same types of tumor taken from different patients possess widely divergent combinations of aberrant genes. This observation presumably reflects the many different routes that individual tumors can take to escape the cell's normal anti-tumor protections. These findings can be displayed and where mutant genes identified in a large number of colorectal cancers are shown in peaks with a two-dimensional mutational landscape. The height of the various peaks reflect the frequency with which the particular gene is mutated in this particular type of cancer. It is evident from this type of display that a small number of genes are mutated at a large portion of tumors. This can be thought of as mountains in the landscape. For the first part, there are oncogenes that cancer researchers have been focusing on over the years. Each type of cancer has its own characteristic complement of frequently mutated genes. In the case of human colorectal cancer, the three most frequent mutated genes are APC, KRAS, and TP53 tend to become mutated or distinct stages in the progression of this type of cancer. So, mutation in both copies of the APC gene are found in over 60% of the smallest benign adenomas of the colon, suggesting that mutations in this gene can represent a first step in the formation of colon cancers. Larger adenomas, as well as cell masses in the early stages of malignancy, tend to maintain mutations in one of the RAS oncogenes, namely KRAS. In contrast, the TP53 gene tends to mutate or become epigenetically silenced only at late stages along the path when the tumor is clearly malignant. Then, this malignancy tends to contain mutations in one of the RAS oncogenes, namely KRAS. In contrast, the TP53 gene tends to mutate or become epigenetically silenced only at later stages along the path when the tumor is clearly malignant. cells displaying chromosome instability, which is reflected in increasingly abnormal karyotypes, only appear after mutations in KRAS. How a small number of genes tend to be mutated at relatively high frequency in colorectal cancer. However, a surprisingly large number of genes are mutated at lower frequency, less than 5% of cases. However, a surprising large number of genes are mutated at lower frequency. These have identified as HILs. There are approximately 50 different genes representing HILs in mutational landscapes of colorectal cancers. While we can presume that the genes that are mutated in high frequency, the mountains, are important factors in driving cells into malignancy, considerable debate is centered on the roles of genes that are mutated at lower frequency, the HILs. Many of the genes represented by the HILs almost certainly have a causal role in determining properties of the malignancy phenotype, even if they provide the tumor with only a small selective advantage. Mutations that cause or contribute to the malignant phenotype are described as drivers. Other genes that constitute a HIL may simply represent passengers, that is, genes that tend to become mutated for some reason but have no effect on the phenotype of the cancer cell. It may be a daunting challenge to determine which one of these genes are derived and which are simply passengers. In addition to the genes of the mountains and hills, there is a large number of other genes that show up in a mutant state at very low frequency in a population of tumors. Mutations of this class are seen as small circles. It is evident from this and large number of other studies that in other types of cancers that the mutational landscape of the cancer genome is highly complex. Mutational genome landscape of the cancer genome is highly complex. A closer analysis of the mutations that make up the mountains and hill suggests that most of these large number of genes encode proteins that participate as components of a largely small number of pathways. In one study of a person with pancreatic cancer, one of the most deadly and least treatable types, recurrent mutations were found in both more than 60 genes, but for most part, these mutations affected a core set of 12 cellular pathways or processes. Most importantly, half of these pathways or processes were disrupted in 100% of tumor samples. In a comparable study of glioblastoma, the most common form of brain cancer, the majority of tumors exhibited mutations that affected three major pathways: P53, PRB, and PI3K. As discussed elsewhere in this chapter, cancer can be thought of not simply as a disease of aberrant genes, but not as one of aberrant cellular pathways. Mutations in one of its number of genes disrupt the same pathway that things have same consequences for the cells. The PI3K pathway is breast cancer can be activated by mutations in the catalytic subunit PI3CA, amplification of the growth factor receptor HER2. Mutations in the PI3K affect PKB, AKTI, or loss of the PI3K antagonist PTEN. A few of the most prominent pathways that tend to be deregulated in any different types of cancers. The Tumor cancer pathway disease rather than a genetic disease generates more optimism among drug developers because it suggests that disrupting or destroying any of these key steps in a single essential pathway may be beneficial to retrain the malignant cells and lead to major tumor regression. Before leaving the subject of cancer genetics, it should be noted that the view presented here, that cancer is a gradual multi-step progression of individual point mutations, is not universally shared. Some researchers argue that mutations rate in humans is not high enough for cells to accumulate and mutations necessary to become fully malignant during an individual's lifetime. Instead, they have proposed that carcinogenesis is initiated by catastrophic events that lead to widespread genetic instability over a relatively small number of cell divisions. Mutations in a gene involved in DNA replication or DNA repair, as occurs in case of HNPCZ, might rapidly spawn cells carrying widespread genetic abnormalities. According to another proposal, cells that have undergone an abnormal cell division and possess aberrant number of chromosomes are likely initiators of cancerous growth. The best way to decide among these possibilities is to analyze the state of the genome in cell at very early stages in tumor formation. And unfortunately, for the interest of both researchers and cancer patients, it is virtually impossible for identifying tumors that they have composed of a small number of cells. By the time the tumors are detected, the cells already exhibit a high degree of genetic detachment, making it difficult to determine whether the gene alteration as a cause of the effective tumor growth. Then some other aspects of gene expression analysis. A technology for analyzing gene expression using DNA microarrays, DNA chips, was described later. A glass slide is prepared that can contain anywhere from a handful of thousands of spots of DNA, each spot containing the DNA corresponding to a single known gene. Any particular set of genes, such as those thought to be involved in growth and division, or those thought to be involved in the development and differentiation of lymphocytes and some other cell types, can be included within the microarray. Once prepared, the microarray is incubated with a fluorescently labeled cDNA synthesized from the mRNA of a particular population of cells, such as those from a tumor mass that has been removed during surgery, or from the cancerous blood cells of a patient with leukemia. The fluorescently labeled cDNA hybridized to spots of complementary DNA immobilized on the slide, and subsequent analysis of the fluorescent patterns tells the investigators which mRNAs are present within the tumor cell and their relative abundances within mRNA population. So studies with DNA microarrays have shown that gene expression profiles can provide invaluable information about the properties of a tumor. It has been found, for example, that the progression of a tumor is correlated with a change in expression of a particular gene. Certain cancers that appear to be similar by conventional criteria can be divided into subtypes that have different clinical features on the basis of their gene expression profiles. Gene expression profiles of an individual patient's tumor can reveal how aggressive, how lethal the cancer is likely to be. The gene expression profile of an individual patient's tumor can provide clues as to which type of therapeutic strategy will be most likely to induce tumor regression. The earlier a cancer is discovered, the more likely a person can be cured. This is one of the cardinal aspects of cancer treatment. Yes, the certain percentage of tumor will prove fatal even if discovered and removed at an early stage. It is apparent, for example, that some early stage breast cancers already contain cells capable of seeding the formation of secondary tumors or metastases at distant locations, whereas others do not. These differences determine the prognosis of the patient. It was found in a landmark study of the Dutch researchers in 2002 that the prognosis of a given breast cancer is likely to be revealed in the level of expression of approximately 70 genes out of 1,000 which were studied in DNA microarrays. This finding has had important clinical applications in guiding the treatment of breast cancer patients. Those patients with early stage tumors that display a poor prognosis profile on the basis of their gene expression pattern can be treated aggressively with chemotherapy to maximize the chance that the formation of secondary tumor can be prevented. If gene expression data is not considered, these individuals are not likely to receive any type of chemotherapy because there is no indication using conventional criteria that tumor had spread. Those patients whose tumors exhibit a good prognosis profile might be spread with more deliberating chemotherapeutic drugs, even if their tumors appear more advanced. In recent years, several companies have introduced laboratory tests to analyze gene expression of profile of individuals with breast cancers in an attempt to help guide the best course of treatment for these patients. Even though these prognostic tests have been in widespread use for several years, their validity is still being evaluated in larger clinical trials. It is hoped that gene expression profiles can soon be used to improve the diagnosis and optimal treatment for individuals patients with the type of cancer. Then, new strategies for combating cancer. It is painfully evident that conventional approaches to combating cancer, namely surgery, chemotherapy, and radiation, is not usually successful in curing a person of metastatic cancer, that is, one that is already spread from a primary tumor, because they kill a large number of normal cells along with the cancer cells. Chemotherapy and radiation tend to have serious side effects, in addition to having limited curative value of most advanced cancers. There has been hope for decades that these brute force strategies would be replaced by targeted therapies based on our novel and newly synthesized insights into the molecular basis of malignancy. There are several ways that therapy can be considered to be targeted, and it can be targeted to attack only cancer cells, leaving normal cells attacked. And this can be targeted to a particular protein whose inactivation leaves the cancer cells unable to grow or survive. And it can be targeted to the cancer cells of a particular patient based on their unique pattern of somatic mutations. Although the cure rate for most types of cancers has not improved significantly over the past 50 years, there is a reason to believe that effective targeted therapies will become available to treat many of the common cancers in the foreseeable future. This optimism is based largely on the remarkable success that has been achieved with a small number of targeted treatments that will be discussed in this session. So, even though this success have been scattered among the much larger number of failed prospective treatments, they demonstrate that the concept of targeted therapy is sound. In other words, these discussions can be considered as proof of principle just as important. They give both researchers and biotechnology companies the incentive to spend time and money to continue the pursuit of better cancer treatments. The anti-cancer strategies to be discussed in the following sections can be divided into three groups: those that depend on antibodies or immune cells to attack tumor cells, antibodies or immune cells to attack tumor cells, then those that inhibit the activity of cancer promoting proteins, and those that prevent the growth of blood vessels that nourish the tumor. First, immunotherapy. We have heard of or read about a person with metastatic cancer who were told they had only months to live. Yet they defy the prognosis and remain alive and cancer-free years later. The best studied case of such spontaneous remissions were recorded in the late 80s by a New York physician named William Coley. Coley's interest in the subject began in 1891 when he came across the hospital records of a patient with an inoperable tumor of operable, and this operation could not be performed, tumor of the neck, who had gone into remission after contracting a streptococcal infection beneath the skin. Coley located the patient and found him to be free of cancer that had once threatened his life. Coley spent his remaining years trying to develop a bacterial extract which, when injected under the skin into a tumor, would stimulate the patient's immune system to attack and destroy the malignancy. This work was not without the successes, particularly against certain uncommon soft tissue sarcomas, although the use of Coley's toxin, as it was later called, was never widely embraced. Many studies have confirmed anecdotal observations of the body's capability to destroy a tumor, even when it has become well established. In recent years, two broad treatment strategies providing the immune system has been pursued, like passive immunotherapy and active immunotherapy. Passive immunotherapy is an approach that attempts to treat cancer patients by administering antibodies as therapeutic agents. These antibodies recognize and bind to specific proteins on the surface of the tumor cells being targeted. Once bound, the antibody either kills the cell directly or orchestrates an attack on the cell carried out by other elements of the immune system. Production of monoclonal antibodies capable of binding to particular target antigens was initially developed in mid-1970s. During the next 20 years or so, attempts to use these proteins as therapeutic agents was foiled for a number of reasons. Failed, I guess. Most importantly, these antibodies were produced by mouse cells and encoded by mouse genes. As a result, the antibodies were recognized as foreign and cleared from bloodstream before they had a chance to work. In subsequent efforts, investigators were able to produce humanized antibodies, which are antibodies that are largely human proteins except for a relatively small part that recognizes the protein, which remains mousy. In the past few years, researchers have been able to produce antibodies that have a completely human amino acid sequence. Herceptin is a humanized antibody directed against a cell surface receptor, HER2, that binds a growth factor that stimulates the proliferation of breast cancer cells. So, Herceptin is thought to inhibit activation of the receptor by the growth factor and stimulate receptor internalization. Approximately 25% of breast cancers are composed of cells that overexpress the HER2 gene, which causes the cells to be especially sensitive to growth factor stimulation. Up until the development of Herceptin, patients whose tumors overexpressed HER2 had a very poor prognosis. Their prognosis is now greatly improved. For example, one study of 3,000 women with early large breast cancer reported that Herceptin reduced the chance of occurrences of the disease by about 50% in the 4-year period. Recent trials suggest that survival can be increased by combining Herceptin with other monoclonal antibody, for example, Omnitag, that interfere with HER2 dimerization. Over the past few years, a number of fully human antibodies have been tested in clinical trials against various types of cancers. One of this is called Vectibix, which is directed against EGF receptors, has been approved as a single agent treatment of EGFR expressing metastatic colon cancer. Because it is a human protein, Vectibix remains in the circulation long enough to be administered once every other week. Two other human monoclonal antibodies, Yervoy and Arcera, have been improved and approved for treatment of chronic lymphocytic leukemia and melanoma. Like these two drugs binds to CD20 on the surface of B cells. Active or adoptive immunotherapy is an approach that tries to get a person's own immune system more involved in the fight against malignant cells. The immune system has evolved to recognize and destroy foreign materials, but cancers are derived from a person's own cells. Although many tumor cells do contain proteins that are not expressed by their normal counterparts called tumor-associated antigens or mutated proteins that are different from those present in normal cells, they are still basically host proteins present in the host cell. Active or adoptive immunotherapy is an approach that tries to get a person's own immune system more involved in the fight against malignant cells. The immune system has evolved to recognize and destroy foreign materials, but cancers are derived from a person's own cells. Although many tumor cells do contain proteins that are not expressed by their normal counterparts, so-called tumor-associated antigens or mutated proteins, that are different from those present in normal cells, they are still basically host proteins present in host cells. As a result, the immune system typically fails to recognize these proteins as inappropriate. Even if a person does possess immune cells that recognize certain tumor-associated antigens, tumor evolved a number of mechanisms that allow them to escape immune destruction. Many different strategies have been formulated to overcome these hurdles and artificially stimulate the immune system to mount a more vigorous response against tumor cells. An approach is to inoculate a patient with a protein known to be present in their cancer cells, such as HER2 in the case of persons suffering from breast cancer, or CEA, carcinoembryonic antigen, in a person with pancreatic cancer. If a body can be stimulated to mount an immune response against the protein, that response has been the potential attack to the cancer cells that have that protein in their surface. In most immunotherapeutic approaches, immune cells are isolated from the patient, modified or stimulated in one way to another in vitro, allowing to proliferate in culture and then reintroducing into the patient. For many years, clinical trials using this strategy were disappointing, but recent publications have provided the reason for curious optimism. In many of these studies, a significant minority of patients have exhibited a positive response to the treatment, which means that their tumors are at least the size of events. Why some patients respond while most do not remains a focus of investigation. The next method, inhibiting the activity of cancer-promoting proteins. Cancer cells behave as they do because they contain proteins that are either present at abnormal concentrations or display abnormal activity. A number of these proteins are very important. In many cases, the growth or survival of tumor cells is dependent on the continued activity of one or more of these derived proteins. This dependency is known as oncogene addiction. If the activity of one of these proteins can be selectively blocked, it would be possible to kill the entire population of malignant cells. With this goal in mind, researchers have emphasized a virtual use of small molecular weight compounds that inhibit the activity of cancer-promoting proteins. Some of the drugs were customized and protein nanostructure, where others were identified by random screening large number of compounds that are synthesized by pharmaceutical companies. Once a protein inhibited compound has been identified, it is typically tested for effectiveness against a panel of approximately 60 different types of cultured cells, each originally isolated from a different human cancer. Using a diverse panel of cells allowed researchers to reconstruct the genetic heterogeneity. Next topic, the concept of cancer stem cells. Cancer stem cells. Another reason for the failure to develop more effective targeted therapies may be that the agents are not targeting the appropriate cells within a tumor. This possibly requires further explanation but raises an important issue concerning both the basic biology of cancer and its treatment. Throughout this chapter, we have considered a tumor to be a mass of relatively homogeneous cells. And viewed in this way, all of the cells in a tumor are capable of unlimited proliferation, and all of the cells have an opportunity to evolve into a more malignant phenotype as the result of ongoing genetic change. In recent years, a new concept has emerged, which suggests that while most of the cells of a tumor may be dividing at a rapid rate, they have relatively limited long-term potential to sustain the primary tumor or initiate a new secondary tumor. Instead, a relatively small fraction of the cells scattered throughout the tumor are responsible for maintaining the tumor and promoting the spread. The special cells are called cancer stem cells, and there is considerable experimental evidence for this existence in leukemia, brain tumor, breast tumor, and other cancers. In most cases, the evidence for the existence of human stem cells rests in the finding that only a subpopulation of cells in a given human tumor is able to initiate the formation of a new tumor following injection into a susceptible immunodeficient mouse. Those cells that are able to regrow the tumor involve the inoculated animal often possess certain surface proteins that distinguish them from the cells that make up the bulk of the tumor, and these markers that serve to define the stem cell. The term cancer stem cell does not suggest anything about the origin of the cells, whether from a tissue cell, stem cell, or other type, nor does it imply that cancer stem cells necessarily resemble normal tissue stem cells. Instead, they are defined simply as cells capable of propagating or regrowing the cancer. That's it. The concept of the cancer stem cells is raised at this point in this chapter because it has important consequences for cancer therapy. If it is true that only a small subpopulation of cancer cells in a tumor has the capability of continuing the life of that tumor, then developing drugs that rapidly kill off the bulk of a tumor has mass but spares the cancer stem cell is doomed ultimately to fail. This is presumably the reason that patients with CML must continue to take Gleac to keep their diseases in remission. Then last method, inhibiting the formation of new blood vessels, angiogenesis. As a tumor grows in size, it stimulates the formation of new blood vessels, a process termed angiogenesis. You might have heard about this, but if not, please listen to this video. So, blood vessels are required to deliver nutrients and oxygen to the rapidly growing tumor cells and to remove their waste products. Blood vessels also promote the conducts for cancer cells to spread to other sites in the body. In 1971, Judah Folkman and Harvard University suggested that solid tumors might be destroyed by inhibiting their ability to form new blood vessels. After a quarter of a century of relative obscurity, this idea has been translated into an approved anti-cancer strategy. So cancer cells promote angiogenesis by secreting growth factors such as EGF that act on the endothelial cells of surrounding blood vessels, stimulating them to proliferate and develop into new vessels. A variety of angiogenesis inhibitors has been developed by biotechnology companies, including antibodies and synthetic compounds directed against integrins, growth factors, and growth factor receptors. Preclinical studies on mice and rats suggest that angiogenic inhibitors might be effective in stopping tumor growth. Most importantly, tumor treated with these inhibitors in preclinical studies did not become resistant to repeated drug application. Tumor cells become resistant to the usual chemotherapeutic agents because the cells are genetically unstable and can evolve into resistant forms. Inhibitors of angiogenesis, however, target normal genetically stable endothelial cells which continue to respond to the presence of these agents. Inhibiting angiogenesis in human tumors is not easily accomplished as might have been expected based on studies with mice. To date, the most promising results have been obtained with this antibody called Avastin that directed against VEGF, and some other types are also used. For the foreseen future, the best anti-cancer strategy is early detection. The time that elapsed between the initiation of one of the most common epithelial cancers and its progression to a heavily metastatic disease is generally considered to be at least a decade and often much longer. Consequently, there is ample time to detect these cancers before they become life-threatening. A number of screening procedures are currently in use, including mammography for detecting breast cancer, pap smears for detecting cervical cancer, PSA determinations for detecting prostate cancer, and colonoscopy for detecting colorectal cancer. It is hoped that advances in proteomics will eventually lead to the development of new screening tests based on the relative levels of various proteins present in blood or other bodily fluids. This approach is very important, and they might reveal the presence of cancer, including mutant DNA, particular mRNA, abnormal carbohydrates, and distinct metabolites. Screening tests based on each of these types are being type of biomarkers are being investigated.